Hi everyone, I'm Neel. I'm going to be talking about how we can train universal 3D segmentation networks that generalize across imaging platforms and target organisms and don't require any form of retraining or new annotations. For context, for each new project, a biomedical practitioner first acquires their images and then generally wants to perform statistics on segmented key structures. They then have to annotate these structures to generate training data for segmentation networks. However, biomedical images can have millions to billions of pixels, and no busy expert has the time to densely annotate these volumes, especially while trying to maintain 3D consistency. Further, once annotated, the biomedical practitioner now has to train a segmentation network on their limited supervised pairs, which is difficult to make performant. Or they can find publicly available pre-trained networks and use transfer learning, domain adaptation, self-supervised learning, and more to help improve performance. However, while this is straightforward for a machine learning expert, all of this requires the biomedical expert to have machine learning expertise and training infrastructure to execute these pipelines correctly. And this is not guaranteed in practice. And most crucially, this entire process needs to be redone for each new project, which is highly time consuming for both the practitioner and the community at large. The emerging work seeks to remove some of these barriers. For example, SynthSeg, a recent domain randomization method for the semantic segmentation of the brain, starts with a large-scale set of expert segmentations, from which it then samples from an appearance model to generate highly variable and crucially non-photorealistic training image label pairs with much more variation than is contained in all existing neuroimaging datasets. They show that a single semantic segmentation network trained on this data can then out of the box generalize to all forms of neuroimaging across modalities such as CT, various forms of MRI, and generalize across resolutions as well. This works because neuroimaging has a highly standardized imaging setup. For example, patients are typically lying on their backs in the scanner with their heads centered in the field of view. Further, this work uses a vast amount of expert annotations from which it generates synthetic images. And as a result, this type of approach is very specific to the chosen organ to segment. To those ends, our work aims to train a general-purpose biomedical instance segmenter that generalizes to any new imaging modality and platform across all organisms and organs. This network then removes the need for biomedical experts to retrain or adapt the networks on their own for new projects and datasets. As public and instance annotated datasets are especially rare in 3D, and several imaging modalities and organisms have not yet been made available to the public, we propose a method that doesn't use any real images or labels. And to focus the scope of our work, we aim to predict instances matching a common shape prior of interest. To choose this prior, we note that several structures of interest in biomedicine, such as nuclei, nodules, metastases, and others, all have irregular blob-like shapes. So in this paper, technically speaking, we choose a star-convex shape prior to model these structures. Our key contribution is a data engine that procedurally generates virtual image label training pairs appropriate across applications in radiology and biomicroscopy. Our engine incorporates wildly variable appearance models, imaging physics simulators, and models for the image backgrounds and environments. And once such a data engine is constructed, we train a single network on its outputs to segment any biomedical Star-Convex instance at test time without any retraining. At a high level, we first sample a variable number of instance labels with blob-like shapes. We then assign each blob a Gaussian with randomly sampled moments and sample from an overall Gaussian mixture model to generate the foreground. We then multiply it with Perlin noise to simulate spatial texture. We will describe the background later in this talk. Once sampled, we then use an extensive geometric and intensity-based augmentation pipeline to generate wildly variable appearances and geometries. And we repeat this process at random for each training sample to generate images reminiscent of various forms of microscopy and radiology applications. Our augmentation pipeline uses both geometric transforms shown in pink and intensity transforms shown in blue. It uses a wide variety of imaging physics simulators, and its order of application and each of its components' hyperparameters is handcrafted in such a way to generate useful training images. For more details about these decisions, please see the paper. For the image backgrounds, they can often be darker than the image foregrounds, as in various forms of fluorescence microscopy. In other applications, the foregrounds and backgrounds can have highly nonlinear relationships between them. And often, the foreground star-convex instances are embedded in very complicated environments with all sorts of textures with variable shapes. For this, we use another procedural renderer for the background that is detailed in the paper. Ultimately, we train on virtual data reflecting all of these assumptions and refer to this model as AnyStar mix in our experiments. We also train on each of these individual assumptions alone as ablations. Our segmentation network that is a unit that predicts objectness and distance maps used to parameterize Star-Convex predictions. We use the Stardust 3D work to do so, and we further tune it extensively for our application with details in the paper. We find that once trained, the network learns approximate invariance in its outputs, shown on the right, the significant imaging variation in the synthetic input images, shown in the center. For our experiments, we assemble a collection of highly variable biomedical datasets, including fluorescence microscopy of C. elegans in P. dimerili nuclei, mouse cortical nuclei in micro-CT, zebrafish brain nuclei in scanning electron microscopy, and bold MRI of the in utero human placenta. We find that a single network can segment relevant star-convex instances across each and every one of these datasets, all without ever seeing them during training and without ever fine-tuning or adapting them at test time. In comparison to other generalist models for cellular and nuclear segmentation trained on purely publicly available real datasets, such as CellPose, we find that for datasets where CellPose has seen real images from these modalities, the proposed model, shown in orange, trained on synthetic images, performs comparably. However, on modalities and tasks that CellPose has never seen, we find that the proposed method performs much more closely to the ideal dataset-specific reference performance shown in blue, which is a fully supervised network trained on that specific dataset. As a highlight from our ablations, when segmenting dark cortical nuclei in micro-CT, a model trained on randomized foregrounds and backgrounds is able to zero-shot segment instances of interest as expected. However, a model trained on virtual data with bright foregrounds and dark backgrounds fails catastrophically out of distribution, highlighting the importance of intensity randomization. Finally, the final model, trained on a mixture of all the ablation models, improves performance further in regions indicated by the white arrow. Our work does have its limitations. For example, unlike brains, there is no canonical image size in general biomedical imaging, and our results are sensitive to the choice of image size. We train on 64-cube image patches and then apply sliding window inference on larger volumes. There may be significantly better multi-scale methods to improve inference on unseen sizes. Further, our network is trained in such a way that it will segment star-convex instances of any intensity. In applications where only some intensities are of interest, our networks will produce spurious predictions. Fortunately, these can then be filtered out by simple post-processing. In summary, in contrast to proposing a new architecture or loss, we focused on synthesizing training data to train a generalist biomedical instance segmentation model. To that end, we developed AnyStar, a domain-randomized data engine with a carefully designed stochastic appearance and shape model to simulate variable biomedical environments. We then showed that a single network trained on the data engine can then segment unseen biomedical datasets without any form of retraining. Our code, pre-trained model, and interactive Colab notebooks are all available at the link below. Thank you for your time and for listening.